this is going to be the last session of today. I know a lot of people had to go for curfew reasons or childcare duties, but great if you're still here with us. So this is all we have to teach you in terms of the hands-on session. But now you are expert tractographers, but what we haven't actually taught you yet is what the method is, how it works in detail, and what you can and cannot do with tractography. So this is part of the last session. I'm going to do the first lecture where I tell you the do's and don'ts of tractography, and then Laurent Petit is going to take over and show you where we can take it in the future by trying to combine Klingler dissection, postmortem, and in vivo tractography dissections a bit more. Right. Now you know how to dissect your tracts, how to play around with it, how to use it as an anatomical tool, as a segmentation tool, how to bring in functional activation maps to see how that matches or maybe doesn't match the structure connectivity underneath it. And now what I want to talk to you about is, what is the method? What's the method based on? And what are the do's and the don'ts of tractography? So here we go. We have seen that studying the brain on the surface and the connections, you can get those very pretty pictures, but now it's time to learn how to actually get to the point when you don't have Michel or me feed that data to you. So first of all, it's not one method, there is many different methods. And there have been many challenges also as well in recent years of the method. So what you see here is just a collection of the plethora of tracking algorithms and software packages that you can use to get your tractography ready for dissection. Now, I'm not going to say one is better or the other, but go out there, read the literature. Depending on what it is you want to do, one might actually be better than another. But that is up to you to keep on top of what the latest recommendations for your specialized area are. Now, regardless of the tracking algorithm and software you use, there's basically 3 types of tractography that we distinguish. And the first one is deterministic tractography. So deterministic tractography basically is a whole brain tractography where you just segment out the connections between parts of the brain that you want to see. The second one is known as probabilistic tractography, and that is the method where you have a seed and a target region and you isolate the connections between those two, but you get a probability or a reproducibility between your seed and your target. And then the last type of tractography is fantastic tractography, but we only use that one for brain art competitions. So do not put that in your scientific publications. Right, so that's basically all it is. 2 types really, just saying it's 3, it's 2 types. But how do we do it? How do we get there? Well, we already mentioned it a couple of times that it's based on diffusion weighted imaging. And the idea behind diffusion weighted imaging is often explained with that famous drop of ink in water. So the idea is you drop a drop of ink in water, and over time, you see how that ink of water distributes in your, that drop of ink distributes in your water. And you can see that the more time you allow it to diffuse, the further it will diffuse. And we can actually measure that mathematically. So that is really the fundamental idea of diffusion imaging. You just tag water molecules and you see where they end up. Now, if we bring that back to the brain, you have areas where you have high and low diffusivity. Now, this was briefly mentioned yesterday, I think, by Marco Düring's talk. Basically, you have areas where there is very little restriction to movement of water molecules. For example, in the ventricles, and that means that water can freely move around. There is no restriction, and it will, over time, move equally in all directions. Now, the other possibility is that you have areas where you have restrictions, and that could be gray matter nuclei, that could be cellular membranes, but there is something that hinders the diffusivity of water molecules in one direction, and they tend to move preferentially along one axis. So these are the two different types of diffusion we can see in the brain. Now, we can measure that, and if it's free diffusivity, or in the ventricles, for example, you get a sphere reconstruction or isotropic diffusion. And if it is preferential along a direction and hindered perpendicular to it, you get anisotropic diffusion. Now, based on that, we can then create various different maps. So I'm showing you here the mean diffusivity map, the principal eigenvector, the famous FA fractional anisotropy map, and then a color-coded FA map. And from these maps that you get from the diffusion-weighted imaging, we then go one step further to actually get the tractography reconstruction. And the fundamental idea is rather simple. So you zoom in onto the map, and for every voxel, you get an estimate of the diffusion in that voxel, and that could either be along one direction or equal in all directions. And then from voxel to voxel, you just piece them together and you connect it, and that will give you the streamlines that you can see. So that is very much the fundamental idea of diffusion tensor imaging. Now with that, you've seen the slide before, you can dissect out the three main types of connections in the brain. Classically, as I mentioned before, the association pathways are shown in green, the projection pathways are shown in blue, and commensal pathways are shown in red. Now, what's important to know is that this was not invented by neuroimagers. This classification actually goes back to the 19th century, and in particular, Theodor Meinert, who already classified the association, commensal, and projection fibers. So really what neuroimaging and tractography have added to that classification is fancy colors. Now, using that method, we can then track the entire tractogram of the brain. Tractogram just means all the connections in the brain. And you can segment out, as you've just learned how to do, the various different structures that you're interested in. So, tractography in a nutshell, when fibers or streamlines run in parallel, for example, in the mid-sagittal part of the corpus callosum, there's no crossing, they all neatly align, then DTI works beautifully, as you can see on the top left here. Everything is running in line. However, when there is crossing, merging, splitting, the tracking algorithm often fails. And that is because for every voxel, you only get one orientation. So what happens when you hit a voxel where fibers are mingling, crossing, merging, and splitting is that, assuming this is one fiber connection and this is another one, they cross in a voxel. And the algorithm comes to that voxel and suddenly doesn't know where to go because it can only follow one orientation, right? You can probably see that better. So what the algorithm for DTI classically does is three possible things. It either reaches that point and it doesn't know where to go, doesn't know it this way or this way, so it just stops there. That's known as a false negative. So we know there should be connections this way and this way, but we just don't see them. That's what I've shown you earlier with the cortical projections of the corpus callosum, for example. The other option it has when it comes here is like, it doesn't know where to go. So it just creates an average and it goes this way. Now that is known as a false positive. We know there should not be a connection, but we're going that way anyways, okay? The third option is that it reaches that point and one tract is, hang on, let's do it this way. One tract is much stronger than the other, and that just, the winner takes it all. So it follows that direction, but it misses this one. Getting confused seeing myself in the mirror. So to show that to you in a more mathematical way, rather than just with my arms, you see a map here in the top panel. You see for every voxel the DTI reconstruction, and we usually call that the Smarties because it looks like Smarties. And you can see shown by the red arrow, the corpus callosum, they all nicely follow the trajectory and we get a crisp reconstruction. However, if you look at the yellow arrow, we're now in that part where the corpus callosum comes out, the corticospinal tract is coming up, and you have association fibers running along as well. So suddenly there's a lot of crossing, and what you see is that that Smartie in those boxes around that region is not anisotropic anymore, but it turns into a sphere. So in that case, we don't really know where to go anymore. And that is what you see on the bottom panel in figure C. So you see the corpus callosum in red, and you see the corticospinal tract in yellow. And that's exactly what happened here. So the corpus callosum is coming out, the corticospinal tract is coming up, and they cross, and the algorithm doesn't, hang on, here we go, you can see me. And the algorithm doesn't know where to go. So the corpus callosum does not cross through the corticospinal tract, but it stops. So we only see the mid portion of the corpus callosum, but not the lateral projections. Now, with advanced methods, we're actually able to resolve the crossing a lot better. And you see that in the map on the top, where the crossing is resolved to a certain degree. And then also in the reconstruction, you can now see on the bottom that we suddenly see those beautiful lateral projections of the corpus callosum. So this is a huge advantage of advanced tractography methods. And why is that? Because when you have crossing at different angles, the tensor, as I said, has to have one orientation. So it kind of merges them, or has false positive and false negative reconstructions. But if you use an advanced method, the fiber response can be resolved for the crossing and also for lower angle crossing. So not only 90 degrees, but also down to 50 degrees in this example. So the question is, if that is the case and we get beautiful reconstructions, why don't we use it all the time? Why do we still use DTI? One reason is that the FACT algorithm, the classical DTI algorithm, is actually licensed for the clinic, so that's why it's very often used in the clinic. It's also faster and easier to acquire, and we're going to come to that in a minute. But another reason is that with advanced methods, you're able to reconstruct more fiber orientations, but that also means that you have more chances of getting it wrong. So very advanced tractography algorithms, if you look at the tractogram, it looks a lot more messy, and you will spend more time cleaning up your dissections because you can reconstruct a lot of the crossing, but you also go wrong a lot of the time. So what I'm showing you here is an example where I placed a region of interest in the anterior part of the brain, the frontal lobe, where the corpus callosum genome is coming out and is actually crossing with ventral connections like the IF-off and anterior thalamic reconstructions you can see nicely. But there's also a lot of spurious fibers about, especially the one where you see it's crossing over to the splenium to the other side in the occipital cortex. And there's two more examples that just highlight that point. So I want to spend some time now and talk you through what to do to get the best tractography possible. And that will depend on many different aspects that are feasible, but the first one is that you should really optimize your acquisition. The better your acquisition, the better your tractography. The other thing is that you always need to check your data quality. Don't blindly trust your data, and I'm going to hammer that point home to you for the rest of this talk. And also optimize your pre-processing parameters. So that's all the things you can do to get the best data. Once you're at that point where you want to track it, the better you know your white matter anatomy, the easier it will be to dissect it. You need to learn where to place your regions of interest. There's a couple of rules that you can follow, but there's many very useful atlases out there that show you where to place the region for which tract and why. Manual dissection still is the most reliable based on our experience, and it's worth checking the literature regularly to make sure that you use the best and most recent tools. So let's go into a bit more detail. What you see here is cardiac gating. So this is the pulsation of the brain based on your heartbeat. And you can see if you just, I don't know, let's take the bottom panel in the middle. You see the corpus callosum sometimes is touching and sometimes it's not touching. So if you don't correct for cardiac pulsation in your acquisition, then your reconstruction will be sometimes connected and sometimes not connected. So that's quite an artifact that you can easily get rid of by just putting a probe on your participant's finger. The other thing to consider is that the world around us is sharpening up, and so are our neuroimaging tools. The important thing is that some structures are only visible in high resolution data. And I'm going to show you a beautiful example of that. So if we look at the brainstem, often referred to as the Mickey Mouse ears by neuroimagers, you have 4 examples here. The first one is a classical clinical 3T 2 millimeter acquisition, obviously in vivo, and it's quite pixelated, that image. So in terms of identifying fine detail, you're not going to get that with 2 millimeters. A classical clinical 1.5 Tesla would be about 2.5 millimeters, so even more pixelated. Now, one of the best data sets available to the neuroimaging community at the moment is the Human Connectome Project data, or HCP. And their 3T data comes at a resolution of 1.25 millimeters, and you start seeing some structures emerge that you can only guess to be there in the clinical 3T data. And then obviously not to be recommended for your actual in vivo studies, but you can also do postmortem imaging, where you can go down to resolutions below a voxel and get absolutely stunning images, especially when you look at the last one, the 7 Tesla 0.2 millimeter ex vivo. But obviously that is by definition ex vivo, so we can't use it in human brains for clinical studies for sure. But also the acquisition time is rather long for this kind of data. But this is how far we can push it, and there's a big development towards higher resolution and more crisper data. The other thing you can tweak with is the diffusion weighting. So how much diffusion sensitivity you want in your data. And I'm showing you an example here from our 2018 study, where you have 3 examples of diffusion weighting. So the first one is a B0 image, so very minimal diffusion weighting in there. Then we have a 500, a B value of 500, and a B value of 1500. And you can, without knowing what you're looking at in detail, you can appreciate the differences in the images. But when it comes to the lesion delineation, so this is the same patient I've shown you earlier, you can appreciate that on some contrasts it's easier and on some it is harder. The other reason why you want to tinker with your diffusion weighting in your images is because depending on the b-value, you can resolve the crossing better or less good. So here's the example where you have the degree of crossing from 90 down to 30-degree angles and from 500 to 3000 b-values. And you can see the higher the b-value, the sharper the resolving of the crossing at lower angles. So I said I'm going to hammer this home to you, and we're going to have Bill Nye help me do that. Look at your data. Bill Nye may not be able to save the world, but he might just be able to save your research study. So look at your data. And what do I mean by that? So when you look at the raw diffusion data, you sometimes see artifacts like this one, like a signal dropout that could be due to head motion or other factors, but you sometimes see just one of the volumes missing in your acquisition. You also see head motion, especially with patients. So one little trick I have for you here is, if possible, split your sequence. So that's what we did with my stroke studies and the one I showed you from brain. Patients were not able to tolerate the full length of the scan because they had to move in the acute stroke patients. And we got around that by just splitting the sequence in half and giving patients a break in between, and then afterwards in a pre-processing, we concatenate the data. And there can also be Gibbs ringing, for example. Now, all of these are not detrimental. It doesn't mean you have to throw your data out, but you have to know you have those artifacts in your data to correct for it. So always look at your data. Now, the next step is, it's not just push a button and you have your tractography. There's multiple pre-processing steps to follow. And I stole this little video from the FSL wiki page, and you see on the left the raw imaging data before any correction, and then the iterative steps with applied correction. And if you just compare the first and the last image on the bottom row, you can appreciate that in the first one, you see those black lines, which are missing values. But in the final image, you don't have that anymore. So it does a fantastic job in correcting for artifacts. And then finally, there is, as I've shown you, a lot of variability in the brain. So just be aware of it, be aware of your tracts being left lateralized, right lateralized, bilateral. Be there in children or just in adults, the changes across time, plasticity, like these are all factors you need to account for when you do your tracking. So in terms of best practice, know your anatomy and know your data. Look at your data. Understand the method you're using and its limitations. Where possible, optimize your acquisition parameters, especially in the clinic, in a clinically acceptable time. Account for variability and regularly check new developments in pre-processing to correct your data to the best standards, but most importantly, keep mapping and keep connecting. Now, with that, I'm very happy to hand over to Laurent, who will show us where we can take it next by combining Klingler and tractography methods. Laurent.